Hi everyone. On this talk today, we have JackDiGiovanna, the Program Director at Seven Bridges, and Mark Cowley with the Children's Cancer Institute in Australia, representing the Zero Childhood Cancer Program. And now I will pass this over to our speakers, who will talk about their goals in reducing pediatric cancer to zero.
First, I'd like to start off with a bit of background information that childhood cancer is the leading cause of death in the Western world. And in Australia, that means that there are about 1,000 new cases per year. And in the US, I believe it's 11,000 new cases of childhood cancer every single year. Thanks to decades of research, fortunately 80% of these children survive their current standard treatments, which are generally combinations of chemotherapy and radiotherapy, carefully developed over many years and many clinical trials. So unfortunately, that still leaves 20% of patients with poor outcomes from these standard treatments, and it's these patients that we really want to improve outcomes for. Furthermore, of the patients that do survive their childhood cancer, about 70% of them have lifelong medical implications due to their treatments. And so it'd be great to be able to treat these patients as effectively, but with far less long-term side effects. Next slide, please. Okay, so we strongly believe cancer is a disease of the genome, and shown in this slide on the left is what a normal genome looks like down a microscope. Pairs of chromosomes inherited, one from mum, one from dad. And in the middle, even down a microscope, you can see that a cancer cell can become highly mutated. There's lots of macroscopic changes here, like copy number amplifications and deletions of entire chromosomes. And in the bottom middle are a whole series of marker chromosomes where two chromosomes have become joined together by so-called structural variants. Now, on the right-hand side though, with technologies like whole genome sequencing, we have unprecedented resolution to investigate the cancer genome all the way down to the single base pair level. What I'm showing you is 8 different patients' cancer genomes from the deadliest brain cancer of all, diffuse intrinsic pontine glioma. You don't really need to be a cancer genomicist to see that these 8 different profiles are extremely different. From the ones on the right have lots of copy number changes, those green and red blocks. And the ones on the left really don't have very many copy number changes at all. And each of them has thousands, if not tens of thousands of little dots around the outside, single base pair changes. Every patient's cancer genome is unique, and that's really one of the essences of why it's so hard to treat cancer today. Next slide, please. So this is a great segue to the now reasonably well-established idea of precision medicine, which is where we aim to use as many comprehensive techniques as possible to better understand each patient's tumor and harness that information to treat that patient with the right drug, preferably at the right time, and preferably ultimately earlier on in their treatment journey rather than at the end of their treatment journey, which is where a lot of precision medicine research is happening at the moment. Next slide, please. So this is also a good segue to introduce the Zero Childhood Cancer Program. This is Australia's National Pediatric Cancer Precision Medicine Program. We launched this program in 2015, and it is now a national program. Currently, Zero focuses on children with very high-risk cancers. These are the children that don't really have a very good treatment plan when they are initially diagnosed, or those that have relapsed and progressed through all of the standard treatments known to them. So by the time they're enrolled onto Zero, there's really not many more options available for them in the standard clinical environment. Within Zero, we perform a whole suite of comprehensive molecular profiling tests on the right, including whole genome sequencing, RNA sequencing and methylation profiling, and a range of in vitro and in vivo testing where possible. The goal is to find out all this information about each tumor and return that to the patients in real time. This is perhaps best exemplified by this case of a 10-month-old girl who presented with a tumor almost the size of her chest cavity. And in 9 days, we were able to sequence her cancer genome, identify a new type of genetic change driving her tumor called an NTRK fusion, and make a treatment recommendation to a drug which had only been released a month before this patient was enrolled onto the study. This was 2 years ago, and this patient is now tumor-free thanks to finding the right drug for this particular patient. This is precision medicine in action, really, the combination of genomics, pharmacological innovation, and getting that patient to the treatment, to the patient at the right time. So in a nutshell, does this work beyond the first patient? Absolutely. We've just published our findings that show that in a nutshell, this approach works in many patients. In most of the time, we can find the molecular basis of each tumor. 70% of patients we were able to make a treatment recommendation for, which, as I remind you, that these patients really didn't have any other treatment options before they were enrolled. We found a really high rate of inherited cancer variants, meaning there's potentially a reason in their family why the patient had their cancer. 5% of patients, we changed the diagnosis, which is really important for managing those patients. And ultimately, of patients that received the targeted treatment recommendation, 70% of them had a response, about 30% of those really with tumor shrinkage. So my last slide really is to say that Zero Childhood Cancer is a national program with a huge number of national and international partners. And that's a really great segue to say that cancer is still essentially a collection of rare genetic diseases. There's only 200 cases per year in Australia of high-risk cancer. And it's imperative that we share our data with national and international partners so that we can accelerate our understanding of childhood cancer. This is a great segue for me to introduce JackDiGiovanna, SVP and Program Director at Seven Bridges Genomics, to tell us how we can achieve this. Thank you.
Thanks so much, Mark. I really appreciate the introduction, and that was a great, those are great slides that you gave. My name is Jack. I'm the program director at Seven Bridges. So our team is responsible for all of the work that we do with the federal government, be that the NIH or the Department of Veterans Affairs, and with nonprofits such as the Pancreatic Cancer Action Network and with Zero as well. So I joined the company about 5 years ago, and at the time we had this vision at Seven Bridges of what if we had a single pane of glass? And it really leads to these really cool graphics where you have folks in different continents all over the world. You have different cloud providers, different regions, and how would you bring this together? And now that was very much where we thought we were living 5 years ago. Now we've gotten to the point as a community, both I would say in the research space and in the commercial space, where we're facing this very concretely, where we're facing data sets with policy level restrictions that cannot be moved, or data sets that are simply so large that it's impractical to move them to other locations. So part of what we did here is in 2018, we launched a capability called multi-cloud that would allow through a single UI to be able to compute on the data where it lives rather than moving data around the world. And that's the story that I want to get into as we talk through how we worked with the Zero team and a large consortium of folks to help make this possible. What's shown here is an overview. Seven Bridges were headquartered in Boston. We worked extensively with Children's Hospital of Philadelphia to develop a Keynote platform that sits going under the Kids First umbrella, with most of the compute and processing, compute and storage happening here in US East 1, also with capabilities in US West 1. And then Zero, in collaboration with Biocommons, the ARDC, NCI Australia, have most of their data here in Sydney. So how can we actually bring that data together and aggregate it, as Mark introduced, such that you're not actually shipping data over to the US for processing and shipping results back, but actually combining the data and analyzing where it lives. What I really liked about this consortium, we got together, we just started really in Q1 of this year, not only the technical challenges, which were really neat to show off different capabilities and to stress test things, but also the scientific questions we aim to address were really exciting. So both the practical ones that I put in bold here to start through with this talk, but also trying to set the foundation in our collaboration for deeper and more impactful questions of what makes rare pediatric brain cancer so deadly in the first place. Obviously more than could be accomplished in a few quarters, but a really exciting trajectory to be on and to be a part of. So first I want to introduce the two pediatric brain cancer data sets that were combined here. So the first one is from the Children's Brain Tumor Network. This was previously called the CBTTC. This is about 900 RNA-seq samples that had already been uniformly processed on Keynote with the Kids First RNA-seq pipeline. And they also had available 8,000 whole genome sequencing samples, all within the Kids First data resource. And from the zero side, there were 100 samples with both RNA-seq and whole genome sequencing available. And these were all stored on the National Compute Infrastructure Australia in Sydney. The idea, the processing flow here was that you would take the zero data and process all of that with the Kids First RNA-seq workflow. That would give you gene expressions. Those could be combined with the existing gene expression data that had already been calculated for the Children's Brain Tumor Network data. The aim being that you could cluster on those, do some dimensionality reduction and cluster on those gene expressions to identify brain cancer subtypes. And if any of those were novel and had not previously been identified, the next step would be look in those novel clusters and try to understand the shared molecular drivers. Genome sequencing both from zero and CBTN and process that with the Semantic Color from zero here. In the next slide, I'm going to present just some details of the Semantic Color. It's based on S2 for SV calling. It includes Strelka and Sage for small indels, Amber for via low frequency, Cobalt for read depths, and Purple for purity estimation. So it includes both SVs and CNV components. Shown here is a graphical representation of this workflow in common workflow language. The blue circles are individual tools. So this is, I can't see it at this resolution, but this is Amber, for example, excuse me. Gray things on the left are the inputs or configs, and gray dots on the right are outputs, and the different wires are how they're connected. The tools are larger or smaller depending on the number of inputs that they have. This can also be fully done in text if that's your style, and fully configurable. I just kind of like this representation for our presentations. So if you're not familiar with common workflow language and why we would use that, you might be familiar with WDL or other workflow languages. They're all trying to achieve the same thing. If you have some code and maybe that's local, you could have it in GitHub. Docker is enabling you to capture the environment such that that would be reproducible. Common workflow language is nicely describing the entire set of processing, capturing all of the requirements that would be for the analyses. And if you take the three of these, you have a very nice description. And as long as you bring it somewhere that has the data and executability, you could reproduce the results. This is really key for us because we're both going to be doing the processing both in US East 1 and in AWS Sydney. And we need to be able to be sure that the analysis is fully portable and reproducible. In terms of pipeline implementation, so the prior implementation was not a CWL pipeline. It had about a 5 to 6 hour runtime for 4 GB tumor normal pairs. However, there was egress costs, admin, and transfer time there to bring that result back. The current solution, the optimized CWL1 pipeline, it had about a 3 hour runtime for the samples that have been characterized. But most importantly, as I highlighted in blue here, is that there's no data movement between regions or providers, thus no egress cost or no admin cost to do that. And the results show up in the same UI as you did the analysis. This was all done on Kavatica. As I said in the beginning with the sort of world slide, this was co-developed between Children's Hospital of Philadelphia, the Data-Driven Discovery and Biomedicine Group, and Seven Bridges around 2016, initially bringing together the PNOC and CBTTC data. In 2017, it became a foundational platform for the Gabriella Miller Kids First program, now also including structural birth defect data, again, a rare disease. The question of how Kavatica works, what is the architecture that would allow this to happen? So it's built on the Seven Bridges core infrastructure. There's a whole host of different services here with very cool names that are not so important, but you could imagine, this is data and compute, security, and billing. These are all running on AWS. So there's some people who have the impression that Seven Bridges is an AWS shop. We do like AWS, but at the same time, this is not the entirety of our platform of Kavatica or any other platform. The first thing I want to show here on the right is connecting to other data sets, which we do for example, NIH data sets that we provide access to. The first step that we did with the collaboration with Zero was to connect to the NCI data sets as storage as well. What that looks like to make the connection is shown here on the UI and the video on the right. So we're taking this example, Broad recently put the Nomad data up on open data for AWS. In this case, the user is creating a project, which we'll get into in a few slides, going to add files to that project, and whereas they could easily bring in data sets or files that they've created in other projects, they could just as easily select volumes. They selected this Nomad volume, they're traversing the structure, the directory structure of that volume, and they're going to bring in a VCF and an index file. And now this appears in the project just as if it were any other file. It can have metadata, it can be analyzed, it can be downloaded, whatever you like. It's just instantly available without having to store it or ingress it. Still, that's only part of the solution. So connecting to the data, you still have the issue of egress. So prior solutions brought data to the compute. And if you were on another platform or a Seven Bridges platform prior to 2018, you could run into this situation where you have data in Sydney, and that's fine. You can just create the analysis project in Sydney and you wouldn't have any egress. But as soon as you wanted to combine with data that was somewhere else, you would have a whole lot of egress to bring all of these fast queues into the Sydney project for processing. So how did we solve this? Well, this is when multi-cloud comes into the picture. So what I didn't talk about in the prior slide, left side here. So you have all the different users of Keynote or any other Seven Bridges platform, and they're interacting with the core infrastructure through the GUI or through the API. But in terms of the compute and storage that's happening, we already have that you can do this in US East 1 or US West 1 here. You can create projects there. And in the work that we did with this consortium, we just made this possible in AWS Sydney as well. Projects, so the left panel there is projects. These could also be called sandboxes, workspaces, depending on what terminology you're familiar with, really are the base unit of organization on Seven Bridges platforms. There's an owner of the project. She can include various project members, give them role-based access control, pointers to the files all live within the project, apps, which are tools, workflows, pipelines, R scripts are there as well, and tasks are also run in that project. They're really important for collaboration and interoperability, and there's also some nice admin aspects of the projects where you can set billing groups and distribute costs appropriately. So in terms of making a project in Sydney, this is real-time me just going through the flow here, creating a new project on Keynote. All I have to do is give it a name from the drop-down, take AP Southeast 2, and that quickly, within less than 10 seconds, I have a project created. So because I have that flexibility through this collaboration, now I can use multi-cloud to bring compute to the data. So same situation we had before, where we have some RNA-seq data or some whole genome-seq data available in Sydney, process that there in Sydney. For the data that's in the US, create a project in the US and run the same processing through CWL. You're sure it's reproducible processing of that data in US East 1. And now, as I introduced in the beginning, I want to bring that together with some unsupervised clustering. Now here there's no free lunch. I'm having to combine the results. There will be some egress here of the gene expression. However, it's orders of magnitude less than if you were bringing over the RNA-seq fast queues, or even worse, the whole genome-seq raw data. And there's also situations where you could do this processing independently and not have to combine. So that's some of the impact that we've had with this multi-cloud configuration. Right now, people in the consortium are clustering the results of this federated analysis, bringing together the gene expression data that have been processed from the Children's Brain Tumor Network and also from the Zero data. Here's a representative example using TSNE, which is showing the different cancer types, glioma and other cancer types, clustering together for some cases and not clustering as well for others. Again, with the goal, the next step from here would be to identify the distinct clusters which were previously uncharacterized, and then use the semantic color on that data to further look for drivers. So I talked a lot about specific Seven Bridges technologies here that made it possible in multi-cloud and connected cloud storage. We're also very involved in the GA4GH and other interoperability groups. The one aspect that I really want to highlight as an important next step for this work is the GA4GH passports for auth-Z. What we ran into here specifically was we had some data that was authorized by dbGaP, other data by a consortium whitelist, other data by local DAC, data authorized by EGA. And it really makes it a really complex situation when you're combining all of this data. How does the policy keep up with how to do that? And that's something that we're really excited about as next steps. To quickly summarize, this partnership created access to the largest pediatric cancer research ever generated in Australia, boosting samples more than tenfold without any more additional sequencing or recruitment. Multi-cloud and connected cloud storage were key components in creating a single interface or a single pane of glass to analyze this data across the 2 continents. And what was really exciting was seeing how the technology, data, and workflows developed. One caveat that I wanted to throw out that policy has to keep up, and that's an important aspect that we have to continue to develop technologies around that to make sure that's respected. And for me, the most gratifying part of this was, as Mark really nicely introduced, Zero makes treatment recommendations based on this gene expression in concert with a number of other aspects of the processing. And I really, what's exciting is this much larger data set could improve precision for that aspect of gene expression, and that really has the potential to make a direct impact on patients and their families. I want to acknowledge teams of people who are really critical to this work, from CHOP, from the Children's Cancer Institute, Biocommons, ARDC, NCI Australia, AARNet, Bioplatforms, and 7 Bridges. And thanks for your time. We'd be happy to take a couple questions.